Hey guys, welcome back to a fun and exciting day of genetics. Our topic today is dihybrid crosses, one of my favorite. And if you're like me and you like those kind of like crossword or Sudoku puzzles, this is going to be right up your alley. Okay, so first question, what's a dihybrid cross? It's a cross or a Punnett square between two traits. So the reason that you would do this is because you want to know, as a geneticist, what the likelihood of inheriting seed color and seed shape. Okay, you want to know what's the likelihood of being a green round seed or a yellow wrinkly seed. I don't want to just know what the probability is of you having offspring that are green or wrinkled, but green and wrinkled or yellow and wrinkled. So you need to do a dihybrid cross. And for every Punnett square, when we did monohybrid crosses, you had 4 boxes. A dihybrid cross is actually 4 times larger and contains 16 boxes. Okay, so it's a little more complicated, but as always, if you follow these steps to the T, you should be able to solve a dihybrid cross accurately every single time. So step number 1 in solving a dihybrid cross is determining your parents' genotypes. A genotype this time is going to include 2 different genotypes together to actually make up a total of 4 alleles. 2, let's use A for the first trait, and B for the second trait, whatever they are. So this would actually be one parent's genotype for a dihybrid cross. The 2 alleles for the first trait next to the 2 alleles for the second trait. Okay, and because you have 4 different letters making up your genotype in this case, you have to FOIL to determine what the gametes are. You can't just simply separate and put a capital. You can't just, like on a monohybrid cross, you can't just put A's over here and B's on top, because this is all from one parent. So we need to figure out which letters go next to each of these sides, and that's where foiling comes in place. So let's practice. Okay, so seed color in these plants that we're looking at, black is dominant to white. So since black is dominant, the allele we're going to be using to represent black is going to be a dominant capital B. The allele we're going to use to represent the recessive white will be a lowercase b. Now, with seed shape, round is dominant to wrinkled, so round is dominant, capital R, and wrinkled is recessive, so lowercase r. Now, to find the genotype, you have to read the problem clearly. And this problem says, find the gametes for a homozygous black hybrid round plant. Homozygous black means 2 alleles that are the same that would give us a black color. That will be capital B, 2 dominant alleles. Hybrid means 2 different alleles. So the full genotype for a homozygous black hybrid round plant would be 2 dominant B's and a dominant R and a recessive R. Now to figure out the gametes, you FOIL. The only way that I do this is I physically draw a letter matching the first allele from the first trait with the first allele from the second trait. And I write that down. Big B, little r, big R. Okay? Then I take the first allele from the first trait and match it with the first or the second allele from the second trait. Big B, little r. And now I can go to the second allele from the first trait. Big B, big R. And the last combination, big B, little r. So there you have it, a little bit nicer than I wrote it. These are the 4 possible combinations of alleles you can have when you take and you mix the alleles for black seed color with a hybrid round seed shape. So let's practice some more. Heterozygous black and wrinkled. If this was the description of of this plant's genotype, I know heterozygous black is 2 different alleles and wrinkled is recessive, so 2 lowercase alleles. And I foil big B, little r, big B, little r, little b, little r, little b, little r. So my 4 possible gametes are like that. Now, the next 2 problems, I did not tell you in words what the genotypes were. Instead, I just gave them to you. But you're still going to foil. And if you're homozygous recessive, if you are white and homozygous dominant round, then you will have 4 gametes that are all the same. Little b, big R for all of them. The most complicated of gamete formation is going to be a plant that is hybrid for both traits. So if you're hybrid black, hybrid round, you're going to have one gamete that's capital B, capital R, one that's capital B, lowercase r, lowercase b, capital R, and lowercase b, lowercase r. So each gamete is different in this problem. Okay, that's step 1. Step 2 is to set up your Punnett square. And to do that, you'll put one parent's gametes across the top and the others down the side. Now, when you do this, look back at this. This is going to go over one box. This is going to go over the next box. This is going to go over the 3rd box, and this is going to go over the 4th box. You must keep both of those alleles from each trait together. Okay, and that goes across the top, and the other parent's gametes go down the side. And then you cross the 2 parents' gametes. But you must keep the alleles for each trait together. Okay, you're not going to be mixing them up because they have to make the genotype for that trait. And the only way I can make the genotype for that trait are to keep my B's together and my R's together. And I have to observe the rule of dominance, meaning that within the genotype for seed color, any dominant allele is going to go first in the B's, followed by the recessive B, if there is one. And then I move on to the genotype for the seed shape, keeping any dominant alleles, if they're present, before any recessive. And then I answer the questions. Okay? So let's solve a problem. Let's solve a problem. Cross 2 heterozygous black hybrid round plants. Okay? So let's figure out the genotypes. Heterozygous, black, hybrid round, both of them, which means that when I do my foiling and I figure out my gametes, they're both going to be the same. Over this first box, I'm going to put my capital B, capital R. Over my second box, my capital B, lowercase r. Over my 3rd box, the lowercase b, capital R, and 4th box, both recessive. Now, since the second parent's genotype is exactly the same, I will put these same combinations down the side. I'll give you a second to do that. So after you've written that down, now you cross them. And I always start in the top left box. Bring the B's down together, and then the R's down together. And that's your genotype in this box. B, B, R, R. Okay, move on to the second box. Bring the capital B's down and over, and then you got to keep the capital R first, then the recessive R. The next box, bring the capital B over from the left, the lowercase, the recessive, down from the top, and then your two dominant R's. The last one in the top row, you have a capital B from the left, a lowercase b from the top, a capital R from the left, and a lowercase r from the top, and you get a completely heterozygous genotype. Okay, I want you to take a second and do the second row. You ready? Let's look in the first box in the second row. The B's, keeping them together, we have two capital B's, and then we have heterozygous R's. Next box, we have two capital B's again, and two lowercase r's. Third box, we have heterozygous B's and heterozygous R's. And the last box, we have heterozygous B's, homozygous recessive R's. Okay, make any corrections if you didn't get this, and then I want you to try the next row. Let's do the next row. First box, I'm going to be heterozygous B's and homozygous dominant R's. Second box, heterozygous B's, heterozygous R's. Third box, homozygous recessive B's, homozygous dominant R's. And last box in this row, homozygous recessive B's, heterozygous R's. And go ahead and do the last row. You should have gotten heterozygous B's, heterozygous R's, heterozygous B, recessive R in the second box, recessive B's, heterozygous R's in the third box, and the very last bottom right-hand corner box, all recessive. Now you'll notice that I color coordinated these, and I did so to help me answer the problems, which were to determine the genotypic and phenotypic ratios. I'm going to have to pause for a second, and then we'll go over this when we start the next video.